Hello, welcome to Breakthroughs from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of June 9th. Let's start. First we will discuss a paper published by Randall Chin in Nature on the mechanism of alpha-Ketoglutarate mediated lifespan extension. Why is this paper so important? Mechanism of aging is not clearly understood. Authors show that alpha-Ketoglutarate extends longevity in C. elegans and elucidate molecular mechanism of lifespan extension by alpha-Ketoglutarate. Authors screened normal metabolites and aberrant disease-associated metabolites and discovered that the TCA cycle intermediate alpha-Ketoglutarate, but not isocitrate or citrate, delays aging and extends lifespan of C. elegans by 50%. Alpha-Ketoglutarate response is dose-dependent as shown here. The dilution or killing of C. elegans bacterial food source has been shown to extend worm lifespan, but the lifespan increase by alpha-Ketoglutarate is not due to altered bacterial proliferation or metabolism as shown here. Increasing alpha-Ketoglutarate levels by RNA interference that inhibits alpha-Ketoglutarate dehydrogenase also extends worm lifespan, consistent with a direct effect of alpha-Ketoglutarate on longevity independent of bacterial food. How does alpha-Ketoglutarate increase lifespan? Authors used biochemical approach DARTs in jerked cells and found that alpha-Ketoglutarate binds specifically to ATP 5B subunit of ATP synthase. They also show alpha-Ketoglutarate inhibition of activity of complex 5 in heart mitochondria and decreased ATP levels and oxygen consumption in nematodes after alpha-Ketoglutarate treatment. So alpha-Ketoglutarate binds and inhibits ATP synthase. Since starvation also increases lifespan, authors looked at starvation in nematodes and found that alpha-Ketoglutarate levels are elevated in starved C. elegans. Since mTOR pathway involved in starvation, authors investigated this pathway and found that TOR pathway activity is decreased in human cells treated with octyl alpha-Ketoglutarate. Also, alpha-Ketoglutarate did not cause significantly more autophagy in either ATP2 RNAi or LET363 RNAi worms, indicating that alpha-Ketoglutarate pathway feeds into TOR pathway. So here is the pathway authors propose for the mechanism of alpha-Ketoglutarate mediated increase in lifespan. Next, let's look into the paper published by Scott and colleagues in Cell. Authors provide mechanism of action of CLEC16A. Why is this paper so important? CLEC16A has been implicated in type 1 diabetes, multiple sclerosis and adrenal dysfunction, but its function is not known. Authors show that CLEC16A interaction with proteins in mitophagy pathway. They also demonstrate regulation of pancreatic function by CLEC16A. Let's look at the experimental data. Authors demonstrate interaction between CLEC16A with E3 ubiquitin ligase NRDP1. Authors also show that proteasome inhibitor MG132 treatment to CLEC16A knockout fibroblasts normalized expression in reduction of NRDP1, suggesting that CLEC16A protects NRDP1 from proteasomal degradation. NRDP1 regulates proteasomal degradation of E3 ligase parkin. During mitophagy, parkin moves to mitochondria. CLEC16A knockout increases parkin levels as shown here and were not reduced following FCCP-mediated mitophagy. In knockout fibroblasts, parkin was found in mitochondrial fraction. Parkin ubiquitinates mitofusin 1 and 2 and promote their degradation. Mitofusin 2 expression was reduced in knockout fibroblasts, consistent with increased parkin activity in mitochondria. CLEC16A loss reduced oxygen consumption rate in fibroblasts and could be rescued by NRDP1 transfection. Similar result is also seen in RNAi knockout. So CLEC16A restrains mitophagy via NRDP1 Parkin pathway. Authors generated pancreatic specific CLEC16A knockout mice, and they were significantly hyperglycemic compared to litter-mate controls in intraperitoneal glucose tolerance testing. Also, these mice showed reduced basal and blunted insulin release, indicating impaired glucose tolerance in vivo. Loss of 16A in beta cells displayed an accumulation of vacuolated structures, partially degraded cytoplasmic material consistent with autophagic vacuoles, and rounded mitochondria with dramatically disordered and amorphous structure. So CLEC16A controls mitophagy and maintains mitochondrial function. Thank you for watching Science Breakthroughs from Science.